I'd like to introduce to you one of my favorite speakers, Dr. Boyd Haley. He received his bachelor degree in chemistry and physics from Franklin College in 1963, and from 64 to 66 he served as a medic in the United States Army. He obtained his master's in chemistry at the University of Idaho and his PhD in chemistry and biochemistry at Washington State University. He was NIH postdoctoral scholar in the Department of Physiology at Yale University Medical School. His first academic appointment was at the University of Wyoming in 1974, where he was promoted to full professor in 1983. In 1985, he was hired by the University of Kentucky Markey Center Cancer Center with academic appointments as professor in the College of Pharmacy and the Division of Medicinal Chemistry and in the Department of Biochemistry. He was appointed to the chair and professor of chemistry and biochemistry in the Department of from 1996 to 2005. He retired from the University of Kentucky in July of 2008, and he has lectured throughout the world and testified before congressional committees and the Institute of Medicine regarding various aspects of mercury toxicity and neurological diseases. Please give a warm Academy welcome to Dr. Boyd Haley.
There's a reason for what I've been doing. And a lot of times I'd walk around and people would talk to me, well, I'm chelating with DMPS or DMSA or doing certain things, and they would talk about the pains in their kidney and how these chelators were not working. And of course they hit my first love, and that's chemistry and designing molecules. You look at my entire NIH career. It was designing compounds and making compounds that other people couldn't make. And that's because I'm just an intense little rat when I get in the laboratory, and I work very hard at it. I have a lot of flaws, but laziness and lack of intensity are not those. So anyway, we're going to start. I want to present some essential concepts so that you guys can kind of follow along with what we're going to talk about today. We came in here, and for various reasons, about 2006, I decided we had to make a chelator that would get mercury out of the body. And the main reason for that is you're never going to get your government to cure the problem of bad water, bad medicine, bad dentistry. They're not going to do it. I'm so frustrated with them. You just want to scream. You have to work around them. You have to come up. And I decided the only way I was going to convince people that mercury, and not only mercury, but the whole compendium of heavy metals that are in the bodies of people was the cause of disease, was to find something to cure. And I can relatively say that we've made big steps in that direction. I don't think there's any doubt about that if you know the data and some of it that we're going to produce. Now, some of the things that I would like to tell you today, I can't, because we have certain things involving this compound that are involved in patent applications. And if I say it in public, I lose it. The company loses it. And I would say this is a project that is primarily funded by people in the IAOMT. Tom Colpitts, David Kennedy, Phil Sukel, Rich Fisher, are some of the people that have bought into my ideas and helped me get the money to test this compound. After we made it and said, hey, we got it. Now look, we've got to do all the testing. So I know I forgot somebody to mention because that wasn't the number I had. But anyway, we're now doing this, and we're now getting near the end of being able to get this compound approved as a drug by various agencies. But the one thing that when I started looking at this, I thought we had to do, and that's look at the dietary supplements and get the idea across. You can go in and take all the supplements, all the minerals, and the legal drugs, and they're not going to work optimally if you're mercury toxic, and if you're mercury and lead, if you're mercury and uranium, if you got all those in your body, and trust me, you guys probably would be shocked if you looked at your doctor's data, urinary mercury, heavy metals profile. Americans, I don't know where it comes from, but we've had a lot of people that are very high in uranium, very high in barium, beryllium, thorium, thallium. I don't know where these people find these metals, but you have a lot of them in there. You will not be able to take a medical treatment, eating a good diet. It helps, and I greatly encourage you to do it, but it will not reverse your medical problems if you're heavy metal toxic. It just won't. And so we have to have a chelator. We have to first clean the body up of all the toxic heavy metals that are loaded within the body. And if you look at the stuff coming out, like from Flint, Michigan, if any of you stop there and stay in a hotel at night like me and drink water, I mean, you got your dose of lead for a while. And if you took the wrong glass at the wrong time, if it had been sitting in the pipes for a long time, you get quite a bit. So we need chelators. We need safe chelators. And they got to be designed logically so they work. And this is what we're going to report on today. It's going to tell you how we've done this. And to work effectively, chelators and antioxidants, because the compound we made that's a great chelator is also the best antioxidant ever made, in my opinion. And if you need to get something inside a cell and something that will respond and scavenge hydroxy radicals, there's nothing better than this compound, which we call NBMI. And but to get it in, the key is none of the chelators you know about today, EDTA, DMPS, and DMSA, get inside cells effectively. They might get in a little bit, but not very effectively, and they don't get in to where the mercury and the lead are bound that causes the toxic problems, the prevention of cell repair, etc. So you have to have something that will pass the membrane, and that was our first requirement when I started designing the new chelators, was I had to have something that would go through the cell membrane, cross the blood-brain barrier, and also get into the bone marrow. And we had that. And the binding of the toxic metals must be exceptionally tight to prevent translocation to other organs. One of the major problems you have, and there have been studies on this, where they've killed rats that were slightly mercury toxic by giving them a chelator that bound that mercury out of the soft tissue in the organs and carried it and concentrated it in the kidney, and the rats died of kidney failure. So we have to worry about that. We want something once it binds it, and if you can get a take-home lesson today, NBMI does not reverse your heavy metal toxicity by grabbing the metals and rapidly getting them out of the body. And that's what modern medicine says you have to do. That's what it doesn't. It binds it and almost immediately renders it non-toxic by making an inert complex that your body slowly gets out by the P450 system, the system of normal detox. Okay, so when we, again, one of the other essential concepts, when you go in and you're really sick, you don't want to wait 6 months to get better. NBMI will immediately reduce the oxidative stress in your body. I mean, it reacts very fast in reducing the hydroxy radical concentration because it reacts with it. And in doing this, you build up your glutathione levels, and as you elevate your glutathione levels, your body increases its ability to detox many, many other toxicants. Because in your body, when you have a toxic material like bleomycin or something like that, or Tylenol toxicity, you have to have glutathione to bind to the reactive products to carry them out of the body. And if your glutathione levels are low because you're mercury toxic, then you're building up all other kinds of toxins. This is a very complex study and a very complex issue that you have to follow. And so a low GS level leads to a reduced ability to excrete all toxic heavy metals, and matter of fact, many toxins such as Tylenol addicts that are very toxic. So we have to get rid of that first, and that's what this compound that we're going to talk about today does initially. And the accumulation of toxic metal leads to an even greater level of hydroxy radical production and more oxidative stress or inflammation. Oxidative stress and heavy metal toxicity go hand in hand to destroy the cells and the health synergistically. In other words, if you have a little bit of lead and a little bit of mercury, you're in much worse shape than if you just have mercury or lead alone. They work hand in hand. And so a compound that would enter cells and remove hydroxy radicals, mercury, and the accompanying free redox heavy metals like copper and iron is needed to treat many illnesses. And that's what we went about building. And this is the compound. And I want to tell you, this compound is totally made out of, most of you remember I sold this as an dietary antioxidant. This part of the compound here is benzoate. It's used as a food preservative for beer, candy bars, Cokes, etc. Everybody in this room has had their shot of benzoate sometime today if you ate store-bought food. It's commonly used. It's not reactive. It's not toxic. As a matter of fact, many people will say it's a very good free radical scavenger on its own. I'm going the wrong way. And coupled to it is this compound. This is nothing more than cysteine with the carboxylic acid group moved off here, because if I left it on there, it would make this a charged compound and it wouldn't cross membranes. So the secret here is to make a compound out of totally natural products that are known to be healthy, known to be free radical scavengers, and make an uncharged molecule that can go through the lipid bilayers and enter your cells, cross your blood-brain barrier, etc. Now the other aspect of this is it's got to have the flexibility where these solvers can move around and bind mercury at a 180-degree angle. That means it binds mercury tighter than anything because that's stable. However, it also has a lot of freedom of rotation and movement, so you can bind a lot of metals in here, and you also have this nitrogens up here that can interact with the metal if it's sitting at this site. Pardon me. But anyway, this is the compound we have. It's a very straightforward to make, and we've tested it, binds mercury, and it will precipitate it out of the solutions, and we'll talk more about that in the lecture. The leaching, when this compound binds mercury, pardon? Is it a better one? Oh. If you look at this, if we make an NBMI mercury complex and incubate it at these pHs for this period of time, and look at the amount of mercury that's released or leached, it's 0.05, less than 0.5% over, actually we saw none, but to be within accuracy and say that we didn't overstate our case, it does not let mercury come out over 36 days or 30 days. This compound binds mercury and it's never going to release it. It stays in your body until it gets modified and sent out by the P450 system. And as you can see here, it allows a very small leaching of a very large amount of mercury at this point in time. Now, why this works, and this is something that if you're a chemist, you enjoy these things. This is the compound in a stick model with the right size spacing between the different bonds. And if you look at the sulfur 1, it's like sulfur 1's in my right hand, or my left hand, sulfur 2 is in my right hand. The reason this works for so many different metals and binds the ones that bind so tight, which is, we'll talk about in just a second, all of the metals that are toxic to you, is that this thing can move around, you know, with degrees of rotation. In other words, this sulfur here can go all the way over to this side. It can move over there because it can rotate around these bonds here like this, and these bonds can swing, and it can go left and right, and so can this one. So it can position itself just perfectly to trap a particular metal due to its coordination chemistry, because not all metals, even though you see them, they're all Hg2 plus or Pb2 plus, the electron shell around them is quite different. And so some of them have to be bound, you know, almost from a 45-degree angle, and some have to be a 180-degree angle. And so the coordination chemistry, you have to have a compound that can change and interact with all these different coordination chemistries to bind all of these metals. And that's just a lot of chemistry that a lot of you probably don't want to hear about. But if you look at this, you can see that it does it for mercury, lead, iron, cadmium, copper, etc. It binds all these metals and it removes them from the solutions at a very high percent. And this means that if you are lead toxic, and I say this to emphasize what's going on in Flint, Michigan, these kids are lead toxic. If you want to treat them, this compound would treat them as well as it would remove mercury from their body. And it's the kind of thing we've got to get through the government regulatory aspects to get it checked properly. But it binds all of the metals that are listed on this aspect right here, and at different pHs, and it would essentially clean the body of most of the toxic metals that you are likely to be associated with. Now, what I would tell you, the one that's not on here is arsenic. This compound has a tremendous high affinity for arsenic-type compounds. So, and with binding mercury, you have to worry about these two sulfur groups, but there's some metals that are less associated with mercury and like interacting with nitrogens, and you can form this type of compound with metals that are a little bit different than just mercury alone, and you can chelate things like uranium. We have had really great results with this compound. Totally takes uranium out of the body. I mean, there's none left, at least in the urine. Now, in this particular one, we say, well, it binds iron. This is a very important concept. We're going to talk now about why this metal is so safe. It does not remove essential metals that are bound to carrier proteins, which are almost all your essential metals. They can't be free. Your body does not like having copper and iron free. If it is free, it's toxic. And so we were doing this as a study we did off this side, where we're talking about NBMI binding free iron. And these are iron bronchoalveolar lung cells that are excreted or washed out from people. And you can read it up there. You can say it. You can read it clearer than I can say it. Bronchoalveolar or lavage samples. And then we added iron to see if iron caused the same problem as you see in the bronchial cells. And you can see that iron, when you add vitamin C is an electron donor, and vitamin C can be a pro-oxidant or a pro-antioxidant depending upon the situation it's placed into. And you can see that if we chelate this, leave it alone, that you get some hydroxy radicals, some consumption of vitamin C making hydroxy free radicals. If you chelate that with DTPA, it binds metals much harder than anything I know of, but it kills you. If you put this in a human and you put in too much, it will take all of their essential metals out and they'll die. So you can't use that one, but it works real well on the test tube. And this is one that's going to bother a lot of people. This is what happens when you add EDTA to iron. And under this system where there are hydroxy radicals being produced, EDTA actually increases the rate of hydroxy radical formation. And that's been proven in pure things. If you look at the Fenton reaction and you add EDTA to iron that's catalyzing a Fenton reaction, which is a production of hydroxy radicals in a test tube, the EDTA will make it go faster. But if you add NBMI here on the very end, it totally blocks it. Blocks the production of hydroxy radicals or the consumption of ascorbate. And it does this without producing any toxic effect. And then you'll see later that in animals we can add huge amounts. Now, when you see that, you say, this compound binds iron very tightly, renders it not available. Well, what's it do with humans? Is it going to take the iron out of your body? And this is from the phase 1 human study that we did in Sweden, was done in Sweden. And you can see that over the period of the study, it did not deplete any iron out of the patients that were being treated. Now you say, why is that? That doesn't make sense. But this is a take-home lesson, and every metal that's an essential metal, like copper and zinc and iron, go in the body this way. They're never free. In your intestine, this is kind of a hard angle for me to see, but in your intestine, you get the iron converted to the proper form, it gets bound, and it gets transported into the cell. And in the cell, it gets released and it's bound up, either stored in ferritin or goes to other carrier proteins. They get into the blood and take it to the proper cells where iron is needed. But the key thing to remember is it's never free. Free iron is disastrous to your body. And when we treat people with NBMI, we measured their hemoglobin levels, the iron levels, they never dropped. Same thing with copper. But copper has about the same type of process. It's never released free in your body. If it is, you have Wilson's disease. And here's the studies we've done on free copper. And you can see that NBMI is 20 micromolar copper. And then when you start adding NBMI, when you get down to 20, you've just about totally eliminated copper's ability to generate hydroxy-free radicals. Now, why that's important and why you want to do these things is that there are many people, a school of thought that thinks that the destruction in Alzheimer's disease is caused by free copper. I mean, people publish that, and it's widely followed, and that's because copper can generate hydroxy-free radicals. Now, the difference is, if we give somebody mercury, they go into oxidative stress. In other words, they start making free radicals. But as a chemist, you know that mercury can't do that. If you put mercury in a Fenton reaction, it will not donate an electron to make a hydroxy free radical. That takes what they call redox metals, such as copper or iron, in your body primarily, or zinc, perhaps. But so how does mercury do this? Mercury does this, and it causes you to have oxidative stress in your brain because it crosses the blood-brain brain barrier better than anything without slowing down. And when it gets in there, what's it do? It displaces iron and copper that are bound in the mitochondria. So the mercury goes in, knocks the iron off of the electron transport chain, and we know it does that because it inhibits ATP synthesis. But when it releases that copper and that iron, then you have a Fenton reaction taking place. You have all the components there you need. The substrates are there, vitamin C's there, just like you're seeing in this thing. This is ascorbate produced. But mercury is involved only in a secondary way by displacing the free copper. You can't get free copper in your brain. If somebody starts telling you about you're eating copper and that's the reason you're getting Alzheimer's disease, the copper would never get from your stomach into your blood, and it sure as hell would never make it to your brain. It'd have to pass all the carboxylic acid groups, like in the citric acid cycle, citrate, etc. It just wouldn't make it. It has to go there, carried in there properly. And then mercury vapor is not touched at all. Mercury vapor, you breathe it, goes right into your brain without any trouble at all. And then it gets oxidized to Hg2+, and then it goes to all the sites that bind copper 2+ or iron 2+ and displaces them, and then you have these free redox metals in your brain, and you start producing hydroxy radicals, and you start getting tissues to to fall apart and not work. And also in our phase 1, to keep this going, when we measured copper levels, there was no depletion of copper levels in a normal body with increased use of NBMI. And that's because there's no free copper there for it to bind. NBMI will not go bind metals off of good proteins because the binding of copper and iron and to your normal proteins, to normal binding sites, are very, very tight. This is some research we're doing at the NBMI. It's under patent consideration now, so I can't talk a lot about it. But what we did is we took cells from COPD patients and looked at them. And the first one, you'll see NBMI reduces oxidative stress in lung cells better than N-acetylcysteine. That's what people use today. And there's no comparison. If you look at it carefully, let's see if we get on there. This is 50 micromolar NBMI, and it's brought down. You have to go to 5,000 micromolar NAC to get the same thing. And you can't get that much in your body. It would be poisonous to you at that level. It decreases IL-6 better, which is a measure of a toxic effect, better than N-acetylcysteine, and again, it's a 50 compared to 5,000 micromolar concentration. It takes 5,000 micromolar NAC to match the NBMI effect. And if we look at the oxidative stress, and if you just look at the bottom here, it makes it simpler and easier to use. You can see the NBMI totally blocks it, as good as a DTP, except it's not toxic. This compound would kill you because it gets into where certain iron and copper stores are, whereas the NBMI does not, and it has no lethal, no toxic effects at all, and yet it works incredibly well. Now, this is for those of you that talk about taking vitamin C and trying to get your glutathione levels up. Here's the reason why it works. It's called the Meister cycle, Alton-Meister, between vitamin C and glutathione. What it says that vitamin C is an electron donor, essential to health. Ascorbate can be regenerated from dehydroascorbate by glutathione, reduced glutathione, and conversely, ascorbate can be used to salvage glutathione, reduced glutathione from the oxidized form, which leads into a programmed cell death. So I don't expect you all to remember all this, but you have to keep in mind there is a direct tie-in towards your body's reducing abilities and taking vitamin C. People who give you large doses of vitamin C are really based on logic. It works. It doesn't work as well as taking NBMI because that goes in directly. But there is a tie-in, and if you look at this, one of the key components made here is NADPH. And if you make that, that's used to reduce alcohol. I mean, in alcohol, all the dehydrogenases, like alcohol dehydrogenase, NADH is the substrate that allows you to get rid of toxins like alcohol. And that's a detox system for us. We're not supposed to have alcohol in our bloodstreams, as you know. So we can go back and we talk about oxidative stress. That's just nothing, it's two things. Increased hydroxy free radicals and low reduced glutathione, increased oxidized glutathione, GSHI. And when we have this, the body can't detox. It can't get rid of everything. Every toxin that comes into your body in a steady process, you can't get rid of it unless you have an adequate amount of GSH. And it's both the total amount of glutathione and the ratio of the reduced versus oxidized glutathione that are concerned in health. If you don't have enough glutathione at all, the ratio doesn't help you much at all. But if you have a high ratio, and most of it's oxidized glutathione, your cells are going to go into programmed cell death. And mercury can cause that. Mercury causes the very rapid production of GSH into GSHG, and that's part of the toxic effect, because that now starts apoptosis in that particular cell, and that's programmed cell death. And you can measure this quite easily, and we're going to show you some of the places where we've done this. Total blood glutathione levels are easily obtained. I don't know how accurate it is. I like to talk to doctors. When you send the glutathione, they measure the GS versus GSSG ratio in your blood. And we've done that with people before and after reduced glutathione. And reduced glutathione, as I'll show you in a few minutes, dramatically goes up in people who take NBMI. And the ratio of oxidized glutathione, the level of oxidized glutathione drops off dramatically. It makes a very healthy system then. Okay. Now, for some of you that are more visually oriented, this work was done by Ragnar Klingberg. Some of you met him. He's in Sweden. He's a CEO of NBMI. And, you know, and I forgot to read the disclaimer. I do have a financial interest in the product in my talk and the company that's doing it. And it's called NBMI and it's called Amerimed. But Ragnar and I spent a lot of time early on, especially talking about how do you check these things. And what I wanted to do more than anything, because I had done it in fish and I had done it on a sick cat and everything else, but he told me that they had places in Sweden where eagles come in to be fed. And especially when they start getting sick and they can't fend on their own, they go there and then they fall down and they routinely die there. And there's a group in Sweden, he told me, that took care of these birds and tried to recover them without much success. And so this is the first one that was found. And I don't know if any of you have ever tried to pick up a wild bird, especially one that's got a beak like an eagle. You just don't do it. I mean, unless they're damn near dead. And that's what this eagle was. This eagle, it's a bald eagle, a female, I think. And you can see she's sitting there and they're trying to get her to eat rats. I don't know what stage it was, but she was found paralyzed and unable to eat. And the veterinarian in Sweden gavaged NBMI, I think 1,500 milligrams, 1,200 milligrams over 5 days down the eagle's throat. And then the eagle got up, started eating, and this is the eagle sitting on a perch. And they had it in a soft cage, you know, one of these cages that are made out of screen, so if they hit it, they didn't kill them. And I guess this eagle, when it got healthy, didn't like being in there at all and was turning into a real dangerous bird. And they knocked the door open one day and flew up in a tree, and they flew out. But this was, I think, about 2 months after he'd been in there. It's impossible to recover them. You know, we have a saying on the farm, if you have a sick chicken, you have a dead chicken. Just a matter of time. You can't cure them. I mean, the birds we haven't learned how to cure birds. We know how to keep them from getting sick. But if they get sick, they're just about gone. And this was remarkable that this eagle is now flying around Sweden somewhere. And what this proves, for those of you that why this is important to humans sitting here, is that NBMI is not toxic to predator birds in huge amounts. That's a lot per pound or per kilogram, the amount they squirted down this bird. So it's not toxic to them, just like it's not toxic to rats, cats, and dogs, and humans. And it also tells you, this eagle couldn't fly. He couldn't even hold his head up. And so you know that mercury, he had been mercury toxic for a long, long time. And the mercury had plenty of time to get into the deepest areas of his body that are the most protected. His mitochondria had to be shot, totally blocked, and down there, and he was just a few days from death, or you wouldn't have picked him up. So it tells you that NBMI will go wherever mercury goes to bind it and render it non-toxic. Otherwise, this eagle would not have got up and flown away. And the level of mercury in his blood was over 900 micrograms per liter of blood. In humans, if you're above 10, you're taken to the hospital for infusions, etc. So this recovered a bird that was more than acutely toxic, massively acutely toxic, and for a long time. So this, it says that NBMI works quite rapidly to bring the bird back to good health. I mean, in a few days, he was eating rats again. And but before that, he couldn't be. And so we did the same thing now. We repeated this with an owl that had 356 micrograms of mercury per liter of blood. And that owl, I think, is going to end up in a zoo in Stockholm somewhere. The eagle was too radical or too active to be there. So this is just what I told you, and you can read it. But it's really quite remarkable that you can take an animal that's that far gone and bring him back with giving an oral dose of a chelator antioxidant such as NBMI. So we can say NBMI is an exceptionally good antioxidant that we can use at high concentrations due to its proven lack of toxicity. In other words, you can overdose these birds. You don't have to sit there and say, do I give 100 milligrams or 150? Give them whatever you want. Just make sure you give enough. And you can't kill them by giving them too much until you fill their guts up with it. It can affect the general increase in intracellular reduced glutathione, indicating a significant reduction in oxidative stress. And this protects, in today's society, you hear stuff, we're all low in vitamin D, we're low in vitamin E, etc. And it's not that we're not eating enough, it's because we're more lead and mercury toxic, and this destroys your vitamins in your body by oxidizing them and making them non-effective. And NBMI reverses that. And I know some of you have seen this slide before, and this is a comparative ORAC scores, oxygen radical absorbance capacity. In other words, this is where you look at the capability of antioxidants from diet, from whatever region you want, to say how effective are they. And from this, you can see that NBMI is one of the best antioxidants ever made. More important than that, it gets inside the cells. You have so many antioxidants that in a test tube look real good, like resveratrol from red wine, but it doesn't get inside the cells, so it's not very effective when you take it as a tube. It looks good in a test tube, but in treating people, it just doesn't work that well. And you can see all the rest of these different antioxidants that are promoted and sold to cure people, and you see that they're not very effective. The other thing that's important about this means that when you take NBMI, the vitamin E and vitamin D and the vitamins that are susceptible to destruction by oxidative stress would be protected in your body, because an ORAC measurement is done by measuring the destruction of a water-soluble vitamin E complex called Trolox. And if your compound binds hydroxy radicals faster and a lot better than Trolox, which is really also very sensitive to binding hydroxy radicals, it's going to protect your body. The vitamin E that you take, if you're mercury toxic, is rapidly destroyed by the hydroxy radicals that are in your body. And this compound stops that. Okay. Now what happens with hydroxy, and this is, everybody says, this compound, if you take it, it's so stable that it's going to build up in your body and you're going to die. It's going to collect in your head and your head's going to fall off. I've heard every story. But you're looking at somebody that's been taking this every day since 2006 when I first got the data back from the rat study showing that you could pour this into a rat and you didn't have any effect on him. Even at 30 days at 1,000 milligrams per kilogram body weight, the rats showed no sign of any type of illness at all. And that's when Boyd Haley started testing it, rubbing it on the back of his hands. I'm not an idiot. I mean, I didn't take a lot to start with, but I watched it, and it'll go through your skin is what it'll do if you have it in DMSO. And then I started putting a little bit in my mouth and eating it, and I went up to where I was taking about 200 milligrams a day. And the only change I noticed is that people would, when I was a lot younger, when you guys first met me, they would say, he's a lot more volatile than most people. I mean, I didn't mind telling you my opinion, especially if you made me angry. And it was kind of easy to get me angry from what I understand. But you know how when you walk around and you've got a lot of work to do, people are putting, I was chairman of a department, you have to write letters, you have to write reports, and you got to make meetings, and you flinch in your body and you say, oh my God, did I write that letter? Did I send that report in? I had that all the time. I mean, I was constantly checking myself out. And once I got on NBMI, I didn't have to do that. I mean, I could just think, did I do it? Yeah, I did that. It was gone, just in the flash of a second. It really makes you a lot more calm, and you're much more collected when you have good glutathione levels. It really works quite well in that regard. But, you know, it is a concern. What happens to it when you put it in the body? So we burned up a lot of money people in this room have given me doing experiments that you have to do to keep the FDA happy. And one of them is to put the compound in with rat and human liver homogenates and see what happens. So what are the metabolites of OSR or NBMI? And if you look at this particular thing, what we have uncovered, I'm trying to keep this as simple as I possibly can, is that if you put NBMI, the dimer, in with rat liver homogenates, it gets oxidized. It goes to a sulfate. When it scavenges hydroxy radicals, it's got to convert to something, and it converts to a sulfate. Couldn't be a better answer for us, because if you, we'll talk about, just a minute, let's go. There's the common molecule we're talking about. Now, what happens to NBMI? This molecule over here is totally water insoluble. This is NBMI, but the minute you put an SO3 on it like that, from absorbing 3-hydroxy radicals, you make it water soluble. That's how your body gets rid of benzene. Benzene is totally hydrophobic, and it doesn't go out of your body at all. And the process of getting it by your P450 detoxification chemistry is that you have the benzene, which is hydrophobic, dissolve your skin, your body, and everything else, and it's very toxic. In the phase 1 of the P450 system, you add a hydroxyl group here, and then the phase 2, you put a sulfate on. When you make it a sulfate, that's benzene sulfate. It's not toxic at all because your body can clear it out through the kidney very fast. That's the reason sodium benzoate's not toxic, is because you have carboxyl groups on it. It's negatively charged, it's water soluble, and you can clear it out through your kidney. Now, if you look at this, going back to this molecule, there's a site here where P450 could put a hydroxy radical. So if you buy this, even with Mercury, you still have a site up here that you can modify that and have it carried out by your P450 system. And we've checked that out, and that's exactly what happens with this compound. Now, this is data where we gave NBMI. This was done by Julie Buckley, Dr. Julie Buckley in Florida. She treats autistic children. And we were looking at people. These are older people. And the point to make here is that if you look in the first thing on each one of these sections, you'll see what it was to start with. And then you go out to the outside, you can see how the ratio changed. This is a ratio of reduced glutathione to oxidized glutathione. And the take-home lesson, in case you've got bad eyes and you can't see that, is that all 10 people we had in this study, old as well as quite young, these were young children that were sick and under oxidative stress. You can see that the total level of reduced glutathione goes up dramatically, and these people start detoxing and they get better. This is in humans. So what we were looking at is that, here's one that's probably easier to see. But if you compare just a short treatment, the amount of, the ratio here was 40 and it went up to 80 something 87 from 40 to 87. And the ratio went from 15.7 up to 24.3. So that, and the free glutathione. So what we found out with that, in this study, we took 10 people and we gave them 200, I think it was a little over 200 milligrams per day, and we did it for 3 months. And all we saw were improvements. We measured over 200 biochemical parameters in the body, and the only ones that changed significantly were those revolving around reduced and oxidized glutathione. So what we can go to the end and say, NBMI has a very high oxygen radical absorption capacity, which is what a good antioxidant is supposed to do. Nothing comes close to it. And the ability to penetrate cells to the mitochondria. In other words, mercury gets in the mitochondria. Mercury primarily kills you by disrupting your mitochondria. And how many of you read articles where they talk about, for example, autistic children? They have dysfunctional mitochondria, they'll say. Like it's a genetic problem. It's not a genetic problem. It's a heavy metal toxicity problem, in my opinion. Many of them, although there can be genetic issues also. And taken orally, NBMI has been shown to increase intracellular glutathione and to increase the reduced glutathione over oxidized glutathione ratio, which is something that has to be done if you're going to get well. And this is all proven. This is done in humans, and it's working exceptionally well at this time. And NBMI does this by scavenging hydroxy-free radicals, as we showed on the one thing where it's got a high ORAC score. And then, but more importantly, it not only chelates mercury, it chelates the copper and the iron that have been displaced from their natural binding sites by mercury. And these redox metals, and it stops the production of hydroxy-free radicals in its tracks. It really makes you healthy. It's just like you're fresh out of the womb of a healthy mother. And so this is a cartoon because I was told when I was learning how to teach that some people are very picture-oriented and you should have a good picture of how a compound works. And if you look at this and you see that mercury over here in this thing, as the mercury uncharged goes straight through a lipid bilayer, scientific fact, and when it gets in here, it interacts with an enzyme. One of them, the prominent one, is called catalase, and it gets converted from the non-toxic mercury vapor, which then they say there's nothing toxic in an amalgam because it's all mercury vapor, mercury with no charge. And they're right. The uncharged mercury doesn't do anything, but when it gets in your body, it gets converted to Hg2+, which is incredibly toxic, and once in here, it can collect in the mitochondria and cause a lot of functional problems. If you try to treat this with EDTA, DMPS, or DMSA, they do not effectively cross the lipid bilayer. And the reason they don't is because they're charged. Your lipid bilayers are set there to keep charged compounds from getting in your body unless they're supposed to. And then if they do get in, they go in through a specific carrier. And so you can't use DMSA and DMPS to detoxify mitochondria in a toxic animal. It just won't reach it. It won't get there and it won't do it. And we know our compound gets there because on the outside of the mitochondria is the last step of what they call the urinary porphyrin pathway, where we're making heme. And mercury blocks the entry of porphyrins and the cytosol from getting into the mitochondria where they're used to make heme. And if you take NBMI, you reverse that inhibition. So that tells us that NBMI not only crosses here, it gets over here and it gets inside the inner membrane of the mitochondria. Otherwise, it couldn't bind that mercury and reverse that toxicity, that inhibition. The other thing for people to take home, if you look at ethyl mercury and methyl mercury, your body is very high in chloride. And if you have, I've drawn a little reaction over here, EHG+, if you have a lot of chloride and your blood is high in chloride, it will bind with that and it makes that a neutral compound. That's the reason why some people say, well, how does CH3 mercury, methyl mercury, this positively charged, cross the membrane? If it doesn't cross the membrane as the charged compound, it forms the chloride, the Hg cell that's neutral, and it goes straight across the membrane, and inside the chloride is removed and the compound collects inside the cells. And there's a paper, and you can see this here in the red, you can get it off of the computer, off the internet if you want, or off the IAOMT side, where this guy Sharp did a study, and he took animals and injected them with ethyl mercury, thimerosal. And the thimerosal, what he found was that the mercury level inside the cell was 5.6 times higher than in the blood. And that wasn't the end of it. He then isolated mitochondria and checked that out and found out that the mitochondria mercury level from ethyl mercury given as a form of thimerosal that you used to have in some vaccines, still have it, it went up 1,000-fold. It's 1,000 times higher inside the mitochondria than it was in the blood. And that's all chemistry, and that's, I mean, I do understand that, and I've looked at that. That paper is very solid. And so when they say, well, your blood level of mercury didn't go up very much, your blood level doesn't mean a thing. Your blood level, you're constantly pulling that mercury inside the cell and constantly increasing it in the mitochondria. And that's what, if you look at children that respond badly to treatment, vaccinations with the thimerosal, they'll always say they have dysfunctional mitochondria. Well, why wouldn't they? You've been collecting all the shots they got, the mercury's gone inside their mitochondria that cause that. So this is a general, what was going through my mind when I made this compound. We had to be able to get inside there where the mercury went, track it down, bind it, and get it out of there. And the nice thing about it with NBMI, you don't have to get it out, you just have to bind it. When you bind it, it's no longer toxic. I'll show you that data in just a minute. So here's the other thing that I knew we had to do right away was the safety and efficacy testing of NBMI delivered by subcutaneous injection or delivered by oral lavage or capsules. And the reason we did it both ways is because this is the way you can deliver it, and this is what people are going to expect you to do. And I did this some time ago with one of my graduate students, and I remember this because my fear was I was going to kill these chelators. I thought this would chelate essential metals. It just didn't do it. And so we took rats, and we gave them, on day one, we gave them 100 milligrams per kilogram body weight up to 300. Nothing happened. The rats looked happy. And then day 4, we gave them 200, 300, and 400. We upped the dose quite a bit. This is micromoles of NBMI injected, and we did micromoles because that tells me how much I have to get inside there to get the mercury out. I know the levels that are in there. And on day 7, we noticed something unusual. The rats were very happy, and they were developing show coats. I mean, they looked like, the control had a slight gray color to his coat, which, he wasn't very shiny or anything. And the more NBMI we gave him, the better the coats looked. And the higher level, they looked just a lot better. It looked like you were getting them ready for a dog show or something. And so my lack of patience came through, and I decided, well, I'm going to give them 1,500 micromoles per kilogram body weight, which you did down at the bottom, on the very last one. And I thought, this will surely make them sick or make them throw up or make them do something. And no, they were still happy and climbing and running around, and didn't bother them at all. And you understand, we injected this under a flap of their stomach, subcutaneously right here. And so we gave them a total of 2,700, one of them, or some of them, 2,700 micromoles of NBMI, and if this was something toxic, they should have been dead back up at the 400 micromolar level. And they didn't. We saw no signs of toxicity. This was the very first experiment we had, and that got me very excited. So the toxicity testing was done again. We sent it off. I knew we had a winner. We sent it off because nobody believes the guy who makes the drug. You have to send it to a third-party lab approved by the FDA to test it like you're going to give it, and we thought we were going to give it orally. So we had a commercial laboratory, and they confirmed that NBMI has an LD50 greater than 5 grams per kilogram body weight. What we're telling you, we had no LD50. We couldn't give enough. Their bellies weren't big enough to give them enough to kill them. You could fill up their stomach, and maybe they'd starve to death, but it wasn't toxic. And if you look at that and you stretch it out, this is the equivalent of a 100-pound person taking 227 grams per day, and our treatment range is in the range of 0.2 to 0.6 of a gram. So we're way, way, way below what anybody would say was a toxic level of this compound. And this is a study where we showed that Mice given 1,000 milligrams per kilogram body weight per day for 28 days did not demonstrate any toxic effect. So we have a very safe compound if you're a rat. And then, so we jumped up to dogs, and we had to give them 500 milligrams per kilogram body weight for 28 straight days, and they didn't get sick either. And I can tell you from I give it to my dogs all the time, especially if I see they're getting a slight tremor or coughing. It works great for animals. I'm not trying to sell it to you as a veterinary thing. So in the phase 1 human trial, which is the key one, they're very cautious. This is not our limitation. I would have given those guys, I'd have given them 3 grams each, or 3,000 micrograms or milligrams. But in the process of doing this, we were limited for safety reasons by whatever reasoning regulators use, which I haven't figured out yet, up to 600 milligrams a day, and we did this for 2 weeks, and we didn't detect any toxic effects, nor did we detect any depletion of essential metals in these volunteers. And these were, I think, Swedish college kids. So I can say the lack of NBI toxicity in numerous cells culture was also studied, along with whole animals, and the endothelial cells of bovine arteries and epithelial cells of bovine intestines. This is work that was done I did with Dr. Perinati. Some of you remember him. And several liver cell lines that we used for some of the studies, like doing the modification of NBMI by rat liver homogeneous. It didn't make any of the liver cells toxic either. So then, we knew we had a compound that was safe, that bound mercury toxic, but we didn't know if it would bind mercury or get to the right places in the body. And this is the one experiment that if I ever had a night where I couldn't sleep because I was so excited, it was the night after I got the results of this experiment back. And this is some people would say a Boyd Haley quick and dirty experiment. We took rats and we injected them with lethal doses of mercury, the amount that would kill them. And you can see the very first lethal dose was with, I haven't moved it over yet, I guess. And this is an experiment that's been published, but with no treatment, no NBMI. You can see if we gave 1 milligram, we go over here, and after they started dying about right here, and by the end of this period of time, they were all dead. And then we doubled the dose because we wanted these experiments to see how effective was it. And they started dying on this day, and they were all dead somewhere over in here. This is twice the lethal dose. And then we jumped up and we did about 14 times the lethal dose. And you had to be there, but if you took a rat and you lay him on his back and you pull up a flap on his stem and you put 15 times the lethal dose of mercury, it works very fast. I mean, you don't have to hold on to them anymore. You just put them off to the side. They lay there and they look stunned, and then they start gaping. Then they start going into convulsions, and a little bit later they die. It sounds cruel. But when we did this and treated them with NBMI, I mean, like even on the very lowest dose, when we gave them NBMI, none of them died. And this was given 20 minutes after we gave them the mercury. And we put the mercury on the left-hand side and the treatment, the NBMI, on the right-hand side. So this compound will spread throughout the body and totally block the effects of toxic mercury. And as you can see from this, with no treatment, if we went to double the dose, they died very quickly. And if we treated them with NBMI, they didn't die at all. And then we went up to 14 times, and they were dead. It's a matter of hours, 6 to 7 hours, they were mostly dead. And after you gave it to them, when I gave them the NBMI treatment, they were in convulsions. I mean, the rats couldn't walk, and I just thought they were going to die. But I put them in a cage anyway, and the next morning they were up running around eating and drinking and never lost any significant amount, like a total recovery. So this compound is very effective, but it's effective in that it binds the mercury very quickly, and it renders that mercury into an inert complex that does not cause any toxic effect. And to prove that, I made the NBMI mercury complex in a test tube. I mean, the chemistry on that's very simple. You just add mercury to NBMI, in a water-soluble solution, and it'll precipitate out. It just comes out like snow. And you can wash it with EDTA to make sure that there's no free mercury in there. And we sent this off to Southern Research Institutes with the idea that they would gavage this mercury chelated by NBMI down the gullets of these rats and measure the toxic effect. And not only did they die, if they died, did they have any toxic effects in their liver or kidney. You can see the lesions. Mercury has a real destructive effect on the kidneys. And what happened, none of them died. The NBMI, 2,000 milligrams per kilogram, which is enough if it were just straight mercury by itself, enough mercury in that complex, that it would have killed them in a couple hours. They didn't die at all. They lived for a long time. And then at the end of 5 days, we sacrificed them, and they did the histology on the liver and kidney and internal organs, and you can see no sign of any toxicity at all. So the compound, when the NBMI binds it, that should be solid proof. It just renders it into something that's not toxic. So NBMI prevention of methyl mercury and methyl mercury. This is some work we did with Ohio State University, and I want to tell you that because this is give me a hint when I've been talking too long. Okay. I'll write it too. Okay, I just want to get through this part here. I'll go very fast. Dr. Perinati, and I know some of you have heard his talk, and what he does is he looks at the toxic effects of mercury, methyl mercury, and thimerosal, and things like bleomycin on biomembranes, because that's what mercury really does. If you have you get intestinal problems when you're mercury toxic, it's because your membranes are becoming very leaky. And this is work I did with him, and basically this is a cartoon of a biomembrane, and when you add mercury to that, you see the glutathione levels in these cells. You can measure. They go down dramatically. And these tight junctions, which keeps bad stuff from getting in your body, say from your stomach into your into your blood. And the same thing would happen with the blood-brain barrier. Same thing would happen with the membranes that surround your thyroid gland. We make biomembranes to compartmentalize essential organs and the products that you produce so they can be released at the proper time. And if you do this and look at this, this doesn't mean much to lay people, but this is the control, and there's with the low level of mercury, another higher level, and see, what does it say? No, it's thimerosal. This is methyl mercury, and this is thimerosal. And this is, if you add a huge amount of NBMI to the control, it doesn't do anything. But if you add it to the ones that have the mercury in them, and the thimerosal or the methyl mercury, it totally protects them from becoming leaky, etc. Now here's some easy slides to talk about. This is on the cells. We can measure the glutathione levels. This is control. This is control plus NBMI. And what this tells you, in control cells that are living, you're doing experiments on, that if you add NBMI, they live longer. Their glutathione level almost doubles. If you add methyl mercury to it, it goes from here down to here. If you add thimerosal, it goes from here down to here. But if you have NBMI present, this one didn't drop at all, and this one didn't drop all the way down to where it's dangerous. And so, as far as keeping the reducing equivalents in cells and culture, this compound works as predicted. Very effective. It keeps it'll make your internal. And when this was sold as OSR, the major response I got from the mothers of autistic children was that their stomach aches and their stomach pains ended, and they could now go to the bathroom under controlled conditions without it being too painful. And I think the reason is that because it stopped the leaky membranes in the intestines and in the stomach. One more. This is on DNA synthesis, and you can see that NBMI added to controls doesn't have any effect on DNA synthesis. If you add methyl mercury, you totally inhibit DNA synthesis, which means your cells can't repair themselves, can't replicate. And if you have NBMI present, the damage is not nearly as much. And this is the same thing. What's impressive when your pediatrician tells you thimerosal is not that toxic. Well, compare it to methyl mercury. the aerosol totally wipes out the ability for DNA to replicate and repair itself. In this particular experiment anyway. So what we can say in the conclusion, and I'll quit here, okay? Animal studies on safety and the efficacy of NBMI to treat mercury toxicity showed that NBMI injected or given orally at extremely high doses showed no toxic effect. The single dose, and these rats that we gave them multiple lethal doses of mercury, we were only given one treatment with NBMI. It prevented toxicity and death. One dose of NBMI. And that has now been kind of supported by that eagle study, the eagle and the owl that we found that are toxic. This compound goes in your body, it stays there, half-life of 22 hours if you're a mammal, or a human anyway. And so it says here, and it's very protective for quite a period of time. And I can tell you that NBMI does this by entering the cells of all the tissues. Every tissue we checked had NBMI in it. If we gave it to a rat orally and then sacrificed a rat and measured in his brain, his bone marrow, his fat is higher in his fat tissue than anywhere. I think it goes there and it sets and it kind of leaks out and helps you for a long time if you're fat enough. And it does this by entering the tissues and binding mercury and rendering it into a non-toxic complex. And this compound is doing well. The studies that we've completed so far are very positive. And unlike other chelators, it does not remove the essential metals that you need in your body. It doesn't top them at all. And I have taken this compound, like I said, since 2006. And other than the fact that I'm getting old, and my legs don't stand up here very well like they used to, I think I'm fine. Okay, that's all I have if you want.